All right, so in this video clip, we're just going to go through and review some of the basic information that we talked about karyotypes. So remember, a karyotype is how we analyze an individual's chromosomes. So it's that picture image that has all those chromosomes lined up, and we can basically end up being able to kind of conclude a few things from that analysis. So first thing that we want to do before we kind of jump into a little bit of that analysis piece is being able to define the difference between an autosome and a sex chromosome. So I want you to take a second right now just to remember what these two terms are. So if we're going through and remembering, so if we go ahead and define first what a autosome is, autosomes are the chromosomes listed from pairs 1 through 22. So remember, in a karyotype, you can see 1 is significantly bigger than chromosome at location 22. So we organize them by size. And what I also want you to notice at each one of these locations is why I have it in there that it says pairs at each of these locations, because each of these pairs represent those homologous chromosomes. So at each one of these sites, there are 2 chromosomes. When you inherited from your biological mother, the other from the biological father. So then at site 23, this is where we have our sex chromosomes. So our sex chromosomes will always be at that 23rd pair. So you've got to remember that. And then we're going to kind of jump into differentiating between that biological sex. So next thing I want you to do is take a moment to pause and remember what each sex chromosome represents. So if you're looking at that karyotype, take a moment now to remember what what XY represents and what XX represents. So hopefully we remember this. So XX is female, XY is male. So those are the 2 different types of biological sex we talked about when it comes to karyotyping an individual. Now, if we're just overall looking at an individual karyotype, so something we always want to make sure we're recognizing is the amount of chromosomes that are present. So remember that we said that there are 23 pairs. So if there are 23 pairs, I want you to go ahead and answer for me, how many total chromosomes does that mean are present? So if there are 23 pairs meaning 2 at every single site. So we would just do the simple math of the diploid cell variable, which is 2n. So 2 times 23 would give us 46. So for this individual, so this is a diploid cell. Diploid just means homologous chromosomes are present. These are in our somatic cells. Those are our body cells. So there are 46 chromosomes present in this diploid. If it was haploid, we'd cut that in half, and that's where that 23 would come in. Now, where we're going to transition into next is just being able to examine karyotypes that are atypical. So along the lines we went through real quick and kind of just did the basics of identifying biological sex. And we just did on that last slide talking about how 46 chromosomes are the typical number that are present in human diploid cells. So if we go ahead and just pause, or you examine this right now, how many chromosomes are present in this karyotype? Hopefully you identified 47 total chromosomes. So that is 1 more than what is typically in human karyotypes. And if you notice, there's 1 extra chromosome right now in one of the biological or one of the sex chromosomes. So with that, if we're thinking there's an extra chromosome at this site, would this be a monosomy or trisomy condition? This is going to be a trisomy condition because there's 1 extra. So tri meaning 3, there's 3 sex chromosomes. So trisomy can be at any of these locations, but we want to remember tri, root word 3. So we have an extra chromosome in our karyotype. Lastly, the biological sex of this individual. So think back to what we've kind of said in the past about how to determine biological sex. This individual would still be basically presenting as an anatomical male because they have the Y chromosome present. And if that SRY gene is present on that Y chromosome, they're going to basically express the male anatomy. So we would determine this person is a biological male, even though they have two X chromosomes, which two X chromosomes are typically only seen in females. But since we have that Y present, that's why we're going to determine them as a biological male. If we do one more atypical chromosome or karyotype, if we go through and take a look, I want you to determine how many chromosomes are present here. And hopefully you said 45, because in this particular example, if we look at location 21, we only see one chromosome there. So since there's one chromosome there, so one is missing, is this a monosomy or trisomy condition? This is monosomy. So root word mono meaning one. So we're missing one. There's only one chromosome at a designated location. So we call this a monosomy 21 condition. So the biological sex of this individual is male. Because the Y chromosome is present, technically because this atypical karyotype is focusing on basically a chromosomal mutation at the site of 21 that doesn't impact that individual's biological sex, but we still want to practice being able to identify individual's biological sex no matter the karyotype. So this really quickly walked through the basics. If you still have questions, make sure to go ahead and ask your teacher in class, so that way you can kind of go through and make sure you have all the components that you need to go through and identify when looking at a karyotype. Thanks, guys.